Hello, fellow Catamounts. My name is Todd Kepler, and my future scanning project is in the health and medicine sector with a focus on medical innovation. Today, we're going to talk about some major milestones in medical innovation. First, we have the introduction of germ theory. This was groundbreaking discovery back in the day made by two brilliant scientists, Louis Pasteur and Robert Couch. Before their work, we didn't really have an understanding of the true cause of many diseases, but thanks to them, we now know that many diseases are caused by microorganisms, or germs as we often call them. And this discovery didn't just change our understanding of disease, but it also had a huge impact on medical practice. With the knowledge of germs now, medical professionals started introducing sterilization techniques and hygienic practices in the healthcare setting. And this saved countless lives and continues to be a fundamental part of medicine today. I'd like to highlight a pivotal moment in medical history, the breakthrough in diabetes treatment. In 1921, two scientists named Frederick Banting and Charles Best made a discovery that would change the lives of millions, honestly. They discovered insulin, a hormone that plays a key role in regulating blood sugar levels. This was revolutionary in development and treatment and management of diabetes. And really, before this discovery, diabetes was a life-threatening condition with no effective treatment. But with the introduction of insulin therapy, people with diabetes could lead normal lives. This discovery was nothing short of a miracle for diabetic patients, significantly improving life expectancy and quality of life. The significance of insulin cannot be overstated, really. It has transformed the lives of people with diabetes, allowing them to manage their condition and live healthy, productive lives. It's really fascinating to think about how far we've come since that point. In 1928, a scientist named Alexander Fleming made a discovery that would change the course of medicine. He discovered penicillin, the first true antibiotic. This was revolutionary development that transformed medical treatment by providing effective cures for bacterial infections. And the impact of penicillin was profound, really. It was introduced, marked the beginning of antibiotic era. Before this, bacterial infections often led to serious complications or even death. But with penicillin, doctors were effectively able to treat infections, reduce mortality rates, and this truly altered the course of medicine. And it's really kind of amazing when you think about how one discovery can have such a large impact. A monumental discovery in the field of genetics, the discovery of DNA's double helix structure. In 1953, two scientists named James Watson and Francis Crick made a discovery that would become the basis for modern genetic science. They discovered the double helix structure of DNA. This was a revolutionary development that transformed our understanding of genetics. The significance of this discovery was truly profound. Understanding DNA structure opened up a new field in genetic research, and this has led to advancements in our understanding of genetic diseases and has paved the way for development of treatments of these diseases. A historical surgical achievement was the first successful heart transplant. In 1967, a surgeon named Dr. Christian Barnard made a medical history by performing the first human heart transplant. This is groundbreaking development that really marked a pivotal moment in medical history. The impact of this achievement was pretty big because it broke through and paved the way for development of advanced surgical techniques and the establishment of organ transplantation as a common medical practice. Another turning point in medical history was the identification of HIV and AIDS. In 1981, the medical community recognized HIV/AIDS, signaling a turning point in understanding and responding to the epidemic. This was a moment that shook the world, honestly, but it was also sparked a global response that demonstrated the power of scientific collaboration and human resilience. The discovery of HIV/AIDS led to worldwide public health initiatives. Scientists and researchers around the globe came together to study this new virus, leading to the development of antiretroviral therapies. These therapies have saved countless lives and continue to be a crucial part of managing HIV/AIDS today. Additionally, this discovery spurred extensive research into virology and immunology, opening up new fields of study and leading to breakthroughs in our understanding of human immune systems and how viruses operate. So while the identification of HIV/AIDS was indeed a daunting challenge, it also led to significant advancements in medicine and has shown us how through science and collaboration, we can turn the tide against such epidemics. A significant achievement in the field of genetics was the completion of the Human Genome Project. In 1990, a monumental project began that would span over decades. This was the Human Genome Project, an international effort to map all the genes in the human genome. By 2003, this project was completed, revolutionizing genetics and biomedicine. The significance of this achievement is pretty big, it's pretty immense. The completion of that project has facilitated advancements in genomic medicine, personalized medicine, and our understanding of genetics, predispositions to diseases. It's like having a roadmap, honestly, of human biology that guides us towards more precise and effective healthcare solutions. The breakthrough of stem cell research is also something we want to focus on. Stem cells have been a hot topic in the medical field for quite some time, and these cells have the unique ability to develop into many different cell types in the body during early life and growth. And in addition, in many tissues, they serve as sort of an internal repair system, dividing essentially without limit to replenish other cells. The ongoing advances in stem cell research have shown great potential for treating various diseases, including neurodegenerative disorders like Parkinson's and Alzheimer's. And imagine being able to treat these diseases with cells from your own body. That's the potential we're looking at with stem cell research. But it doesn't stop there. Stem cell therapy is being explored for its ability to regenerate damaged tissues and organs. And this offers new possibilities for regenerative medicine and treatments for conditions that were once thought irreversible. The development of CRISPR-Cas9 technology is also a significant development in genetic engineering. In 2012, this technology was developed that was known as CRISPR-Cas9. It enables precise and efficient gene editing. Imagine being able to edit genes like editing text in a Word document, and that's really the power of CRISPR-Cas9. The impact and future potential of CRISPR is really immense. It's being used in various fields, including medicine for gene therapy, agriculture for crop improvement, and biotechnology for genetic research. It's really like having a Swiss Army knife for medical field. As I mentioned, and developed in 2012, this technology utilizes the Cas9 protein to make precise cuts in DNA, acting like molecular scissors guided by RNA for targeted gene editing. This has opened up new possibilities, including medicine, agriculture, and biotechnology. One of the main advantages is its ease of use, cost effectiveness, and high efficiency across various organisms. This has made it a preferred method for genetic engineering. However, like all technologies, CRISPR comes with its own set of challenges. Ethical concerns such as off-target effects and ethical issues regarding germline editing, and some challenges that need to be addressed as we continue to explore the potential of this powerful tool. The development of TALENs is another significant achievement in genetic engineering. TALENs, or transcription activator-like effector nucleases, are a type of genetic engineering tool that uses customized proteins to bind specific DNA sequences. This allows for precise DNA cutting and editing, much like a pair of molecular scissors. One of the major advantages of TALENs is their high specificity and flexibility. This makes them suitable for targeting complex genetic sequences. However, despite these advantages, TALENs are more complex and costly to design compared to CRISPR, which presents barriers to the widespread use. Despite these challenges, though, the development of TALENs has opened up new possibilities in the field of genetic engineering. It truly is amazing to think about how these tools are shaping the future of medicine. There's also the development of zinc finger nucleases, or ZFNs. ZFNs are a type of genetic engineering tool that uses engineered zinc finger proteins to target specific DNA sequences. This allows for targeting DNA cleavage and editing, much like a pair of molecular scissors, so very similar to the others. One of the main advantages of ZFNs is that they were first developed gene editing technology, providing a foundation for later technologies and offering flexibility in target selection. Despite these advantages, ZFNs come with their own set of challenges, though. They require custom design for each target, which can be time consuming and expensive. Additionally, there's a risk of off-target effects similar to CRISPR. Despite these challenges, though, the development of ZFNs has opened up more possibilities in genetic engineering. Gene editing technologies, CRISPR, TALEN, ZFNs have truly revolutionized genetic research by enabling precise genome function studies and regulatory mechanism exploration. These tools are like a pair of molecular scissors, allowing scientists to make precise cuts in DNA and enabling target DNA editing. These technologies are pivotal, developing treatments for genetic disorders. CRISPR is leading the clinical trials for things like sickle cell and beta thalassemia. Imagine being able to treat these diseases at their genetic root. That's the potential we're looking at with gene editing technologies. However, with great power comes great responsibility, as they say, and each technology faces scrutiny over ethical concerns, particularly in germline editing, raising careful considerations for the approach. As I mentioned, gene editing technologies such as CRISPR have revolutionized genetic research. It's being used in clinical trials. It's also being used for cancer research and engineering immune cells to better target and destroy cancer cells. And the future advancements include reduced off-target effects so that their usage is a lot safer. TALENs have been used to correct disease-like mutations in cells from patients with single-gene X-linked SCID, moving towards preclinical and clinical stages. This is a promising development in the field of gene therapy, and researchers are also exploring the disruption of HIV-1 proviral DNA with the human genome as a potential cure strategy. So this could lead to a game changer in HIV research. And looking ahead, there are challenges to overcome. Enhanced delivery systems for TALENs aim to increase efficiency and reduce potential immunology issues. And these developments are set to further revolutionize the field of medicine. And as we talked about earlier, the ZFNs have revolutionized genetic research as well. These have been used in HIV trials to disrupt the CCR5 gene in T cells to resist virus entry. And this is promising development in the field of gene therapy. Genome editing in stem cells has opened up possibilities for potential therapies in monogenetic diseases like Huntington's disease. And imagine being able to treat these diseases at their genetic roots. That's the potential we're looking at with these gene editing tools. Looking ahead, there's obviously going to be challenges to overcome in efforts to refine ZFN architecture to improve specificity and efficiency, with applications also expanding into agriculture genomics. These developments are set to further revolutionize the field of medicine and agriculture. Gene drives harness the power of CRISPR technology to spread modified genes through wild population. What's the aim here? To control diseases like malaria. Imagine being able to drastically reduce or even eradicate disease-carrying insect problems. The impact on public health could be significant. But as with all powerful technologies, there are important ethical and environmental considerations, and the use of gene drives involve ethical debates and ecological assessments to manage potential risk associated with altering ecosystems. It's a reminder that the science is fascinating. It's equally important to tread carefully and consider the broad implications. So as we continue to explore potential of gene editing, we need to be thoughtful and about the importance and have responsible science. Gene editing in organ transplantation is also something that's pretty exciting in this space. Researchers are using CRISPR to modify pig organs to make them more suitable for human transplantation. This could potentially solve the organ shortage crisis. Imagine being able to drastically reduce or even eradicate the waiting list for organ transplant. And that's the potential we're looking at with the gene editing technologies. This application could revolutionize organ transplantation by reducing rejection rates and expanding the donor pool. However, as I mentioned earlier, with great power comes great responsibility, and extensive testing and regulatory approval are required to ensure safety and efficacy of gene edited organs for human use. It's a reminder that, again, while this science is wonderful, we need to think about the broader implications and risks. Improving crop resilience. It's something that I think is going to be wonderful in the future. CRISPR is being used to develop crops with enhanced traits such as increased yield, disease resistance, and environmental stress tolerance. And this could revolutionize agriculture, making our crops not only more bountiful but also more resilient. These advancements could significantly contribute to global food security by improving crop productivity and sustainability. And imagine having a world where hunger is a thing of the past. That's the potential we're looking at with gene editing technologies. Gene edited crops face regulatory scrutiny and public debates regarding their acceptance. It's a reminder that we do have some concerns to think through. Gene therapy has made significant strides, especially in treating genetic disorders, cancer, and chronic diseases. Through targeted genetic modifications, this is potentially revolutionizing medicine, offering cures and improved quality of life for patients with untreatable conditions. However, gene therapy continues to face ethical considerations and requires careful regulatory oversight to ensure patient safety and equitable access. It's a reminder that while science is available, it needs to be equally important to tread carefully and allow availability to all. Gene editing technologies, as we mentioned, CRISPR, TALENs, and ZFNs are being utilized to target, treat, and correct genetic disorders. This offers hope for effective treatments and has the potential to drastically improve or even cure conditions that previously had limited treatment options. But the journey doesn't stop here. Ongoing research is focused on enhancing the precision and safety of these gene editing tools. And what's the goal here? To expand their use in clinical settings and make these life-changing treatments available to more patients. So as we delve deeper into the world of gene editing, let's remember the profound impact it can have on patients' lives. Advancements in genetic research are improving health outcomes and disease management across various populations worldwide. Innovations such as genome sequencing and personalized medicine are becoming more accessible, allowing for targeted treatments and preventative strategies. However, the expansion of genetic research brings up ethical questions and the need for international collaboration. It's crucial to address equity and access issues to ensure that these advancements benefit everyone everywhere. Let's remember that the profound impact can have on global health. Innovative research directions are emerging, with gene editing poised to unlock complex diseases, enhance precision genetic medicines, and explore new therapeutic areas. These advancements are expected to revolutionize healthcare by enabling more effective and personalized treatments, reducing the burden of genetic diseases. Moreover, the integration of machine learning and AI in gene editing research could lead to faster, more accurate genetic analysis and therapeutic applications. And this is a testament to the transformative power of technology in medical research. Tools like CRISPR's, TALENs, and ZFNs have ushered in a new era of precision in genetic research with profound implications for medicine and agriculture. Looking ahead, we can expect continued innovation to improve specificity and efficiency in gene editing, expanding applications in therapeutics and beyond. But as we embrace these technologies, we must also consider the ethical aspects. As these technologies evolve, we need to ensure that ethical considerations and public debates accompany their application responsibly. So as we conclude our journey through the world of gene editing, let's remember the profound impact it can have on our future. And as we conclude this presentation on gene editing, let's reflect on some key takeaways. Continuous innovation in medical research is crucial for developing new treatments and improving patient outcomes. And it's in this spirit that innovation has brought us groundbreaking tools like CRISPR, TALENs, and ZFNs. But it's not just about the science. Collaboration across countries and disciplines is essential to advance research and address global health challenges effectively. It's this global teamwork that allows us to tackle health challenges on a worldwide scale. Looking ahead, our ongoing commitment to innovation will pave the way for transformative healthcare solutions. As Matt Ridley said, innovation happens when people are free to think, experiment, and speculate. So as we wrap up, let's remember the profound impact of gene editing technologies on healthcare and look forward to the future of continued innovation. Thank you for joining me on this journey.